Dear students, Assalamu alaikum. In the last lecture, we studied renal blood supply and the structure of renal corpuscle. The objectives of today's lecture are to learn about structure of renal tubule, regional differences in nephron structure, and the physiological anatomy of urinary bladder. Please note down the slides as these topics are not given in detail in the textbook of guidance. Renal tubule is the second major part of nephron, first being the renal corpuscle, the details of which we studied in the last lecture. The renal tubule is made up of following parts: proximal tubule, loop of Henle, distal tubule, connecting tubule, cortical collecting tubule, cortical collecting duct, and medullary collecting ducts. This figure shows the basic tubular segments of the nephron. Let's now study the different parts of renal tubule one by one. Proximal convoluted tubule. It is the coiled portion arising from Bowman's capsule. It is continued as loop of Henle. Its wall is made up of a single layer of cells that interdigitate with one another, are united by apical tight junctions, and have a striated brush border on the luminal surface. Loop of Henle. It consists of descending limb, hairpin bend, and an ascending limb. Loop of Henle, descending limb. The descending limb is made up of two segments: thick descending segment and a thin descending segment. The thick descending segment is the direct continuation of the proximal convoluted tubule. Its cells have a cuboidal epithelium with brush border. The thin descending segment is the continuation of the thick descending segment. Its cells are flattened epithelial cells without brush border. I repeat, without brush border. And this segment is continued as the hairpin bend of the loop. That is the U shape of the loop. Loop of Henle, the hairpin bend. It is formed by flattened epithelial cells without brush border. It is continued as the ascending limb of loop of Henle. The ascending limb has two parts, thin ascending segment and the thick ascending segment. The thin ascending segment is the continuation of the hairpin bend. Its cells are flattened epithelial cells without brush border. And it is continued as the thick ascending segment. The thick ascending segment is lined by cuboidal epithelial cells without brush border. And it ascends back to the renal cortex and continues as distal convoluted tubule. There are a few points about the permeability of the loop of Henle that you should be keeping in your mind. The descending portion of the loop and the proximal portion of the ascending limb are made up of thin permeable cells. On the other hand, the thick portion of the ascending limb is made up of thick impermeable cells. Now this fact is of importance when we will discuss the concentration of the urine. This figure shows the diagram of a juxtamedullary nephron. Note the histologic features of cells that make up each portion of the tubule. The length and extent of the loop of Henle. In cortical nephrons, it is short and the hairpin bend penetrates up to the outer medulla. The length is different in the other major type of nephron, which is the juxtamedullary nephron. It is long, and the hairpin bend extends deep into the internal or inner medulla. This figure shows the location of loop of Henle in the cortical nephrons and in the juxtamedullary nephrons. In the cortical nephrons, you can see that the hairpin bend just barely penetrates up to the outer medulla. However, in the juxtamedullary nephrons, the hairpin extends deep into the inner medulla. Another important structure is the macula densa. Now, when the thick end of the ascending limb of the loop of Henle reaches the glomerulus of the nephron, reaches back to the glomerulus of the nephron, the portion of distal tubule, which is between afferent and efferent arterioles, has specialized cells forming the macula densa. The macula densa and the neighboring cells in the afferent and efferent arterioles form the juxtaglomerular apparatus. This figure shows the structure of the juxtaglomerular apparatus. Note the proximity of macula densa cells to the juxtaglomerular cells in the afferent and the efferent arterioles. Distal convoluted tubule. It is the continuation of thick ascending segment of the loop of Henle. It is present in the cortex of kidney. Its cells are cuboidal epithelial cells without brush border, and these cells are called intercalated or eye cells. The collecting ducts. The distal tubules coalesce to form collecting ducts that pass through the renal cortex and medulla to empty into the pelvis of the kidney. From renal pelvis, urine passes through remaining portion of ureter and reaches the urinary bladder. The epithelium of the collecting ducts is made up of cuboidal or columnar epithelial cells without brush border. There are two types of epithelial cells present in the collecting ducts. Number one, the principal cells, and number two, the intercalated cells, the P cells and the I cells. The P cells predominate, they are relatively tall, they have few organelles, and they are involved in water and sodium reabsorption. The I cells are present in smaller numbers. They are also found in the distal tubules. They have more microvilli, cytoplasmic vesicles, and mitochondria. And they are concerned with the acid secretion as well as the bicarbonate transport. In addition to the P cells and the I cells and the cells of the juxtaglomerular apparatus, there are other cells present in the interstitial tissue of medulla which have a secretory function. These are called renal medullary interstitial cells, and they are specialized fibroblast-like cells present in the interstitial tissue of the medulla. They secrete cyclooxygenase 2 and prostaglandin E2. Prostaglandin E2 is an important paracrine regulator of salt and water homeostasis. It is also secreted by macula densa and the collecting duct cells. Although each nephron has all the components described earlier, however, there are some differences depending on how deep the nephron lies within the kidney. So there are regional differences in the nephron structure. The first major type are the cortical nephrons. They are about 85% in number, and their renal corpuscle is situated in the outer cortex. The loop of Henle in these nephrons is short, and the hairpin bend extends to only the outer medulla. Their blood supply is from the peritubular capillaries, and the function is the formation of urine. The other major type of nephrons are the juxtamedullary nephrons. They are about 15% in number, and the renal corpuscle is situated in the inner cortex. The loop of Henle in these juxtamedullary nephrons is long, and the hairpin bend, the U-shaped bend, reaches up to the tip of papilla to the inner medulla. Their blood supply is from the vasa recta, and the function is mainly the concentration of urine along with the formation of urine, understandably. Now, having studied the main physiological anatomy of the kidneys, let's move on to the next topic, which is micturition. By definition, micturition is the process by which the urinary bladder empties when it becomes filled. There are two steps involved in this process, and in layman's words, it is the process of urination. In the first step, the bladder fills progressively until the tension in its walls rises above a threshold level. And in the second step, a nervous reflex called the micturition reflex empties the bladder. To understand micturition and the micturition reflex, it is important to go through the salient features regarding the physiological anatomy of urinary bladder. The urinary bladder is a smooth muscle chamber composed of two main parts. Number one, the body, which is the major part of the bladder in which urine collects, and number two, the neck, which is a funnel-shaped extension of the body. The smooth muscle of the bladder is called the detrusor muscle. Detrusor muscle. Its muscle fibers extend in all directions, and when contracted, it can increase the pressure within the urinary bladder. And the contraction of the detrusor muscle is a major step in emptying the urinary bladder. Smooth muscle cells of the detrusor muscle fuse with one another. And there are low resistance electrical pathways which exist from one muscle cell to the other. Therefore, an action potential can spread throughout the detrusor muscle, causing the contraction of the entire urinary bladder at once. The trigone. On the posterior wall of the bladder, lying immediately above the bladder neck, is a small triangular area called the trigone. And at the lowermost apex of the trigone, the bladder neck opens into the posterior urethra, and the two ureters enter the bladder at the uppermost angles of the trigone. The trigone can be identified by the fact that its mucosa is smooth, in contrast to the remaining bladder mucosa, which is folded to form a rugae. The bladder neck is 2 to 3 centimeters long, and its wall is composed of detrusor muscle interlaced with a large amount of elastic tissue. And the muscle in this area is called the internal sphincter. External sphincter. As the urethra passes through the urogenital diaphragm, there is a layer of muscle called the external sphincter of the bladder. This muscle is a voluntary skeletal muscle. The figure shows the anatomy of the urinary bladder and urethra in males and females. Note the position of the trigone and the two openings of the ureters at the uppermost angles of the trigone. Bladder innervation. The principal nerve supply of the bladder is by the pelvic nerves, connecting with the spinal cord segments S2 and S3. And remember, the pelvic nerves contain both sensory and motor nerve fibers. The sensory fibers detect the degree of stretch in the bladder wall, and these stretch signals are responsible for initiating the reflexes that cause bladder emptying. The motor nerves transmitted in the pelvic nerves are parasympathetic fibers. These terminate on ganglion cells located in the wall of the bladder. So these are parasympathetic postganglionic fibers. And then short postganglionic nerves, they innervate the detrusor muscle. On the other hand, the skeletal motor fibers are transmitted through the pudendal nerve to the external sphincter. So the external sphincter has a separate nerve supply through the pudendal nerve, and these are somatic nerve fibers that innervate and control the voluntary skeletal muscle of the external sphincter. In addition, sympathetic fibers also supply the urinary bladder, and this sympathetic innervation is through the hypogastric nerves connecting mainly with the L2 segment of the spinal cord. And these sympathetic fibers stimulate mainly the blood vessels, and they have little to do with the bladder contraction. This figure shows the innervation of the urinary bladder. Parasympathetic fibers shown in blue are the sensory fibers, while pudendal nerve contains the skeletal motor fibers to the voluntary skeletal muscle of the external sphincter. With this, I end today's lecture. I'll continue with the micturition reflex in the next lecture.